Okay. Sorry, just getting all set up here. This is my first time hosting and doing one, so I will actually be the scientist today. Perfect. Okay, welcome back to Ask a Scientist, everyone. My name is Abby, and you may know I'm one of the educators with the Asheville Museum of Science, and I usually host this segment. But today, I thought it would be kind of fun if I tried to give one of the presentations myself. So today, I will be talking about some of my work in college and even after college with toxicology and endocrine disruption. So we're going to get into what the endocrine system does, and how chemicals can affect it for better or for worse. So my name is Abby. I graduated from biology with a bachelor's in biology, I graduated from college with a bachelor's in biology from Radford University. And then after I graduated, I went and worked at ECU as a research technician for a year, working on a really cool knockout study. It was definitely one of the first of my more serious research projects, and I had a ton of fun and I learned a lot. So I'm also going to bring up some of the methods that I used for that, just because I think they're really cool and it's a huge part of understanding how researchers do their science. So I'm going to go ahead and start by sharing my screen. And so today we are going to be talking about understanding endocrine disrupting chemicals, and I am hoping you guys will be able to see my screen. So I'm going to wait. So there's a lag on Facebook, so I'm just going to make sure it works. And please send in your questions for me along the way. I will be monitoring the chat as well, and I will do my very best to answer them. I will preface this talk by saying that I have been actively out of research for about a year now. So some of this is probably nothing that I'm going to say is super out of date or anything, but there may be advances in some of the fields that I'm talking about that I may not be aware of. So I will do my very best to answer some of your questions, but for now, I'll just go ahead and get started. And so what we're going to look at today is we're going to look at the basics of the endocrine system. We are going to look at EDCs, which stands for endocrine disrupting chemicals. And we're going to talk about my research a little bit. And then some really cool science techniques, as I said. So we'll just jump right in. And so what even is the endocrine system? It's, we have our skeletal system, we have our cardiovascular system, our muscular system, but we rarely talk about the endocrine system. But what it is a collection of glands that produce the hormones that circulate our body, and they regulate all sorts of stuff. So they regulate our metabolism, our growth and development, our sleep, our mood, so pretty much everything. So it starts by releasing some chemicals from our brain that will either activate or deactivate different organs to release certain chemicals. And that's kind of where the molecular biology part kind of plays a role in some of what we're going to talk about today as well. So I don't know if you can see I don't think you can see my mouse, but it's in the red box on this photo to the right, you see the hypothalamus, the pituitary gland. These are really main parts of the endocrine system. But mostly what we will be talking about today and what my research involved was the reproductive development. So we'll be talking about testes, ovaries, and reproductive hormones. So what is endocrine disruption? Endocrine disrupting chemicals are a fairly new topic of study. I would say that Rachel Carson first sounded the alarm in the 1960s with Silent Spring and DDT. But of course the chemicals have changed, evolved, and we've produced a lot more since then. So endocrine disrupting chemicals are any foreign chemicals that's going to interfere with proper endocrine function, so proper development. And we're going to focus today on something called hormone mimics. And one thing to know is that endocrine disruption can be, oops, sorry, there's a phone ringing and I am not going to answer that. There we go. We are going to focus on, you can have natural endocrine disrupting chemicals such as phytoestrogens. So these can be found in plants a lot, like soy products, and we can kind of talk about that a little bit more later. But you can also have synthetic endocrine disruption, so man-made these chemicals that we're introducing into the environment. And a really common, well-known chemical is BPA, or bisphenol A. That's a plasticizer. We see that a lot in our plastic water bottles and, BPS. A lot of products will say BPA free, but really they just replaced it with BPS, which is, just as toxic. So the issue with these chemicals is that they look a lot like our normal body hormones. So you can see that there's a lot of cyclic structure in a lot of these. There's these OH, or these alcohol groups off the end. And the reason that this can be really bad is because, it's because the body can get confused. So these molecules, these endocrine disrupting chemicals, will actually trick the body into thinking that it's a natural body. Endogenous is the word, so it's made naturally in the body, chemical, and it will start reacting with some of our receptors to cause those downstream effects of the endocrine system that I was mentioning in the very beginning. So where are the endocrine disruptors found? We find them all over in pretty much all of our products. So we have flame retardants. So in our clothes and our furniture, they actually spray on chemicals to reduce the chances that it'll catch on fire. We have plastics and cosmetics. So a lot of our water bottles, our food packaging, and our makeups will contain endocrine disrupting chemicals. But today we're going to focus on agriculture. These are the two chemicals that I have the most experience with. So we will start by talking about trenbolone. Trenbolone is an anabolic steroid that farmers use to beef up their cattle to produce more meat. And then we will also touch on vinclozolin, which is a fungicide. So similarly to insecticides and herbicides, a fungicide is just a targeted poison to kill off something else. But of course, they are not so specifically targeted that they're not affecting other things. So this is where endocrine disruption kind of comes in. And so I kind of touched on this before, but why is this bad? So endocrine disruption is bad because the body gets confused. And so on the left-hand side, you can see that I have a picture of the molecule testosterone, which is well known as a male sex hormone. Females have it as well, but males have it in greater quantities. So we have testosterone, and then right below it, we have trenbolone. And I put that in red, so we know that trenbolone is the molecule that is dangerous. This is the molecule that looks a lot like testosterone, but isn't. And so this is bad because our natural hormone, so this red one right here, maybe I should have made the colors different. This natural hormone will bind to our receptors and cause a normal cellular response for development. But then these endocrine disrupting chemicals that look super similar will also fit into some of these receptors. And that's really dangerous because then we're going to have those downstream effects that maybe weren't supposed to be there. So we'll start out by talking about my research question. This was at Radford University. This was my first introduction to research, and it was really exciting for me. My mentor was Dr. Sarah O'Brien. I hope she is watching. Hi, Dr. Sarah O'Brien. So my question was, how does exposure to trenbolone influence the maturation and reproductive development of mosquito fish? And this research was kind of a dovetail from previous research that had been done in Radford's ecotoxicology lab by Emily Guise, who I believe is still finishing her PhD now at UC Davis. But Emily was exploring kind of what trenbolone was doing to fish after they were developed. So my question was, how is this influencing their development from young? So being fry, that's the term for baby mosquito fish, is their fry. So from fry and up to adulthood, how are these fish being affected when they are introduced to something like trenbolone? And it's worthy to note that trenbolone will make it into waterways. So if farmers are using it on their cattle and some farm, it will be excreted into agricultural runoff waters, and it will make it into our ecosystems. This is a chemical like the others I'll talk about today that are found in the environment, and they're found pretty much all over. I know that we found BPA as far as the Arctic Circle. So these are definitely super ubiquitous chemicals and very dangerous. So just to get you oriented, these are Gambusia. So this was my eastern mosquito fish. On the top, we have a female, and bottom, we have a male. And these were really great model organisms to use for this type of research because we're looking at reproductive development. So obviously, males and females will have different reproductive parts. And so they have that phenotype that we would like to observe. And they also look different on the outside. So they're sexually dimorphic, both internally and externally. So we can easily tell just by looking at a tank who's a male, who's a female. And I'm going to discuss a few of the biomarkers that we looked at. So you can see below the fish, they have this anal fin. It's longer in males than in females. You can see that females have a larger body all around, and it's kind of hard to tell, but they do have as well a pregnancy spot. So right behind where it's a little white on their belly, they have a little black dot called the pregnancy spot. And that is characteristic of females. Another reason that they're really good to study is they're adaptive fish with early developmental stages. So they'll grow quickly in the lab, really good for studying development. They sexually mature around 3 months. They're broadly distributed across the world. So we have, mosquito fish are in America, in both the Americas. And they're found in areas that are exposed to trendblown, found in recent studies, and then sexually dimorphic. So they have that observable phenotype. So we'll go over this kind of quickly because this is the more boring part of science that you don't get to see as much. So this was the behind the scenes. I did a lot of animal husbandry, so sexy time for the fish. So what I would do to promote breeding is I would take a male and a female, and I would put them in their own little tank in the corner to kind of set the mood. And it turns out they liked to be in the dark if they were gonna produce some babies. So we actually were putting, we had light timers in the room. So they had a normal day-night cycle. I put construction paper on the edge of my tanks to make it seem really dark. So the fish were maybe less afraid. I think our theory with that was that they felt concealed and they were not at risk. So these fish may not have known that they were not, endangered to predators because they're in tanks in a lab, but they might've just felt more comfortable surrounded by the darkness. So eventually, a lot of them started to produce fry, the babies. So we had two groups. We had a sibling group A and sibling group B from two different males and females, just so we could have genetically similar fry, because you don't want to have too much genetic variation when you're trying to study one thing. But we also wanted to have more than just one rep. So we had a number of different fry. We had our trenbolone concentration as an ecologically relevant dose, which means it's found in the wild at this level. So 10 nanograms per 1 liter was our trenbolone concentration. There's a lot of funny things that go on with this. I actually had a tank completely drain overnight one time, and I lost all my fish. So there was definitely a lot of troubleshooting with this to try and get all of my experimental fishies, but we got there. So what did I look at? A lot of stuff, but we started with the internal analysis. So after, I guess I'll go back to this for a sec. They were exposed for 10 weeks when they were 1.5 months old. So they're not fully developed yet. They, were hardy enough to be exposed to the chemicals, because I don't really know what would happen if we exposed them, right as they were born. So we exposed them when they were 1.5 months old, and they were exposed for 8 weeks. So 2 months of this concentration exposure. So looking at the internal analysis, this is called histology. And this was my first experience with histology. Some of you may know it, some of you may not. But basically what it is I had to dissect all of my fish that I was studying and harvest the organs. So I would take out their testes or ovaries. I would put them in wax, and then you slice them really, really thin. And that's kind of what you see in this picture down here, is you have super, super thin slices of the tissue. And it's called, we use H&E staining, so hematoxylin and eosin. That is a type of staining that will stain the nucleus. Oopsies, what is that? There we go. The nucleus and I believe the muscle tissue around it. So basically I was looking at gonad morphology. Did it change after being exposed? But we also looked at the internal analysis, and I won't really cover a lot of these graphs, but they're just for you to look at if you're interested in seeing kind of what we looked at. But we were looking at the overall influence on weight between males and females, the influence on the size of their pregnancy spots that I had mentioned earlier, body size, all sorts of different stuff. So we were really just looking at the secondary sex characteristics, which are those anal fin lengths, those pregnancy spots, because we want to see, are females presenting more as males after being exposed to this androgenic chemical? So it looks, as I said before, trenbolone looks structurally like testosterone. So if females are being exposed to trenbolone during their development, their body is going to react as if they're being exposed to testosterone. And so that's where the concern comes in, is because what effect will this have on the fecundity of the fish or their ability to reproduce and maintain the population in the wild? What effects does this have transgenerationally? So the fish that, you know, were exposed to it during their development, like if they have babies or try to have fry, what's going to happen downstream of that? So there are a lot of concerns that scientists have about endocrine disrupting chemicals, and we still don't know. They do a lot of stuff that we don't even know still, because they can act on the genome as well. So briefly, we'll go over the histology results, so the tissue results, and looking at the reproductive organs. So here I have our control female, and I have a trenbolone exposed female. And this might not look like anything at this time to you, but we have kind of the eggs on the left of the control female. And then on the right, we see something really interesting. So I'll put this up. This is a control. We can see that our treatment female is starting to really look like our control male. And these are actually seminiferous tubules with sperm growing in them. And so that's super concerning. And we do have one question. Katie asked, you said that these chemicals are in a lot of farming. How much of the chemical would we need to be exposed to in order to see changes to human sex organs? And is that being studied at all? Yes and yes. So scientists are 100% studying the effects. We obviously use model organisms first to understand the basics of what's happening. As I mentioned, EDCs are a relatively new topic, but I will get into some of the health and human effects later in the talk as well, but they definitely are being exposed. And I think that it's confusing to know if this is going to affect humans at the same concentration as it would fish, because we're a lot bigger than the fish. So, and we're not swimming in it. So, you know, the fish were swimming in it. And so this study was looking at a lot of ecological repercussions. So, you know, is the population going to survive? Is it, what is this going to look like in the food chain? How is this, like the loss of mosquito fish, how would that affect the food chain? Stuff like that. But there are definitely effects on humans. The problem is, are exposed acutely, so for a short period of time at a high concentration, or chronically over a long period of time at a lower concentration. So it's really hard to try and understand how a lot of these will affect humans, but we can assume that they do, and that they operate in the same way that they would in an animal, because we are animals. And so that kind of leads me to, why do we care? So there's obviously some health effects that we're going to see. Population maintenance, you know, are we seeing changes that are going to be too dangerous to maintain the population? Chemical regulations, you know, we had the Toxic Substance Control Act, also in the 60s, didn't really do a whole lot. And something to keep in mind is that we share the same environment. So human health connects to environmental health and vice versa, and animal health. I mean, we are animals. I don't even think that should be a separate bubble. But this research is really the reason I became passionate about environmental and public health, and how I kind of transitioned to my work at ECU. So that's kind of what I'm going to jump into now. And so we're going to start, I'll introduce Dr. Krista McCoy. She was my advisor for the year that I was at ECU, and her lab studied a lot of similar stuff as our lab did. They used a lot of similar techniques, but they were really focused on the mechanisms of resilience. So I have this up here. Krista showed this to me one time, and I love the visual. So we have our normal dose response curve. So we have the dose on the bottom, you know, some sort of phenotype on the y-axis. And then you're going to see this transition. As the dose goes up, you're going to see more of an effect on the phenotype, whatever that response is. And so in the middle, the darker blue dots are showing the average, and that's usually what we studied, and that's what I studied in my graphs of looking at the external morphology of my fish. I was studying the average, but what Krista's lab was really interested in is looking at these yellow guys down here. So why are these less of these individuals or these data points less affected than the others. So she was wondering, is there something special about these yellow guys down here that's going to cause them to be less affected, or, you know, save them from whatever issue the EDCs are causing? And so we were studying vinclozolin, which is that fungicide I was talking about. And that one is not androgenic, it's actually anti-androgenic. So it has the opposite effect, where it will sit in the receptor and it will block the testosterone from ever making it there. So it won't cause the same effects as something like trenbolone or testosterone would, but it will basically inhibit the testosterone in males during their development from properly doing its job for reproductive development. And so down here, we're studying the NRF2 pathway is what it's called. So this is just a gene pathway that basically when it's turned on, when we can turn on the gene, it will increase the transcription and translation of detoxification enzymes in our liver. So antioxidant and detoxification enzymes. So a lot of our CYP enzymes, CYP, P450, it's a really specific group of proteins that will help to make, in our case, it was making the vinclozolin more water soluble in the body. So it was making it easily excretable. So the question that we were kind of looking at is, can activating this NRF2 gene increase the antioxidant and detoxification enzymes? And can they do it enough to where we're mitigating the issue, or we're just completely, you know, taking the threat of vinclozolin away during exposure, during development? So I put down one of my PCR gels here. I had to do about 400 DNA extractions and PCRs and electrophoresis, plural, on these animals, because when we were going to test the rescue effect that we had, we could only use our wild type animals or our knockout animals. And so you can see that the wild type animal, the gel, the DNA strip will travel farther, the knockout travels shorter. And then these are hets, or heterozygote animals that have a dominant and recessive gene. So really the reason we had to do so many animals, in short, is because I could only use the knockout and the wild type animals, the animals that had the NRF2 gene and the animals that didn't. So, oh, and we have a question that says, I know the timing of exposure is important. What points of development tend to be the most vulnerable to negative impacts? That's a really good question. And definitely there is a critical period during development where we have these certain floods of hormones. We see it definitely right when we're born and we're starting to develop in those first, I guess for humans, I would say it's like the first 3 years. For mice, it's probably like the first month, maybe not even, probably like the first 2 weeks. And because we were using mice for this study, I didn't even mention that. But there's also, we can also think of it as when puberty hits. I know that there's another flood of hormones there. So it might affect it in that same sort of way during puberty or during adolescence. Maybe when a mom is pregnant with her baby, that's another time that, you know, hormones in the body are meant to be moving around in a different way than your homeostasis line of just, you know, being human. So I think that during those times when the body is readjusting to a new circumstance, whether you're born or growing or whatever, that's the time when you're going to be the most vulnerable to some of these impacts. So this was a really exciting thing for Dr. Krista to look at. So she was really excited to understand if we could activate the NRF2 gene to reduce the impact of vinclozolin during fetal development. And so she hired me as her research technician. This was funded by the NIH, or the National Institute of Health. So we, up here is just a figure I made where I had to essentially I took the fetal mice out of the pregnant mom, or the pregnant dam. I kept my wild type animals. So you'll see NRF2++, that means that they had the they were dominant for the NRF2 gene, or NRF2 minus minus, meaning they didn't. And then out of these fetal mice, I would take their little livers and I would suspend their livers and, well, I would homogenize them. I would mix them all up in all different chemicals. And we had to do steroid extractions on the livers to see if we could see the lessening of the anti-androgenic vinclozolin molecules in the liver real time. So down here, I just have a couple pictures. As I said, my job was to extract the steroids from the livers, identify what molecules were in there, and then quantify how many or how much of that a certain chemical. And I use the liver because the liver is our body's detoxification center. So we were hoping that the liver was going to be our first line of defense in understanding why or how the animals were being rescued from this diseased phenotype. And so I also have a little bit of the molecular biology or the metabolism essentially of vinclozolin on the bottom right. This is just showing all the different pathways within the body that vinclozolin will be metabolized by. There are a few, M1 and M2, these metabolites of vinclozolin are the most dangerous. And then M4 and M5 are actually not super dangerous. So if we could even just convert some of these chemicals to look like these, then maybe we're home free. And so to do this, we detected our metabolites using a triple quad mass spectrometer. I think mass spec is super cool. It's really impressive. It can, detect super low amounts of all these different chemicals. But we did something called targeted metabolomics. So this is where I had a standard to back it up by. So I would, spike in a standard just to make sure, like, this is my signal for my M2 standard. Below, this is a detection of the same N2 transition in a vinclozolin male liver. So this is just no treatment here, just exposed to vinclozolin. And then this is the signal we got for M2, the M2 metabolite in a control male liver, so no vinclozolin. So you can see that this is really messy. We don't have a lot of signal here, but we were able to recover a signal for the M2 metabolite. There's also something that we can do called untargeted metabolomics. This I did not stay long enough to play around with, which I'm kind of grateful, because untargeted metabolomics is really challenging. It's kind of like reconstructing a puzzle. And there's something called the Human Metabolomic Database that you can cross-reference your mass spec data to, which will come back to you in weights. And you can kind of try and reconstruct what molecule you may have based on the fragmentation patterns that are caused by the mass spectrometer. So that's a little hard to understand, but basically these molecules down here will be cut in different ways depending on how they act in the mass spectrometer under a magnetic field and with solvents running through. And so the way that they break apart can be reconstructed after, and that's kind of how we can determine what molecules are there. So it's a little weird. That's why mass spec is super cool to me, but it was definitely a learning curve. And then briefly, I'm just going to share a little bit of our proteomics work. So this I just thought was really cool. I had never worked with proteomics before this. So this was done in PathView. That KEGG PathView was our primary software that we use to look at our proteomic data. So this, we can think of this as a similar thing as we did with the steroid extractions, except it's protein extraction. So instead of looking for trenbolone, testosterone, vinclozolin, whatever it is, instead of looking for those hormones or hormone mimics, now we're looking at the different proteins that were released. So this is, there we go. This is looking at a male, well, so it's actually looking at fold change. So you have to have two individuals to look at this data. So we'll have the red is, when you get up to the red, that's down regulation, so you're suppressing those genes and those proteins. And then the green is upregulated, so that means it's going to be producing more. And so this was one of our metabolism of xenobiotics by cytochrome P450 pathways. So this cytochrome P450 is kind of going back to what I mentioned when the NRF2 pathway was increasing these detoxification enzymes. This is a huge family of enzymes that it could be influencing. So what we're looking at is vinclozolin plus sulforaphane, which is their treatment, and then just vinclozolin. So I didn't actually run any of the proteomics data. I did do some pathway analysis showing me the fold change in the upregulation and downregulation to determine what proteins were made and produced. But this work was really just taking off right as my contract ended. So there's a lot of things that you can look forward to in the field of toxicology. I know that there's a lot of different labs that are focusing on this right now. Co-exposure impacts are one. So what's going to happen if I'm exposed to trenbolone and vinclozolin? So we have an androgenic compound and an anti-androgenic compound. Are they going to cancel each other out and then we're fine? I don't know. We have chronic versus acute exposures, and that kind of goes back to what we were talking about before with the temporal impacts of exposure on development. How long were we exposed? You know, how old were we when we were exposed? What were we stressed out? There's a lot of different factors that could play into how bad the results are. We have fixed versus plastic impacts. So are we able to revert back to a healthy phenotype after we've been exposed? And then synergistic effects. And I do know Dr. Suzanne Brander at Oregon State has some really interesting studies about endocrine disrupting chemicals and microplastics. So we can assume that the microplastics that animals are taking in will also contain endocrine disrupting chemicals. So how that's kind of like a double whammy. So she's studying a lot of the synergistic effects of endocrine disrupting chemicals, climate change, pollution all together. And so that's really exciting, scary, and interesting. And Molly's asking, is there a way to determine what we are being exposed to in our water? Yes, you can get water testing kits. The tricky part is that they're not going to be comprehensive. They're not going to always, tell us exactly what's in our water, especially the less popular chemicals. So I don't think any of the testing strips will tell you if you have trenbolone or testosterone or vinclozolin in your water. They'll test for things like lead or, you know, if you have a well, you'll have different minerals in your well that it will test. Because that's really bad to have too, right? Like Michigan, the Flint, Michigan crisis has lead in their water. And I mean, that's probably the worst example of where we can have endocrine disruption in a human. And on top of that, you have to understand that there are also carcinogenic compounds, lots of them. So something like lead is going to give you, you know, it can give you lead poisoning, cancer, endocrine disruptors. It really has a lot more effects than we understand at this point. So this is the part where everyone's like, okay, now what? Are we just living in diseased water? Yes and no. We can help in some ways. We can ditch the plastic water bottle. So, you know, obviously have a reusable water bottle, have your reusable shopping bags when you go to the grocery store. So just normal things that will reduce the amount of waste that we produce, because when we produce waste, we often have these chemicals sitting in there. You want to clean around your house. So actually a lot of dust around your house can hold these endocrine disrupting chemicals on them. As I mentioned before, they're usually on our clothes or in blankets or toys or just things found around the house. So we often will spray things with chemicals thinking that we're making it safer when really we're not. Eat organic when possible. That's a really hard thing to do for a lot of people. Access to organic food is obviously tricky. Affordability is obviously tricky, but those are going to have a lot less pesticides and fungicides. So I put in parentheses here, vinclozolin and atrazine. Atrazine is an estrogenic herbicide, I believe. Yeah. There's a lot of studies of atrazine on frogs. So that's just another thing amphibians are dealing with right now. So never mind chytrid, they're dealing with lots of toxins as well in their waters. Stop using a lot of cosmetics. So you're beautiful. You don't need the makeup. They have a lot of phthalates in them, BPA, BPS. And then of course you can promote science education. So donate to science organizations, wink, wink. Support funding for research, support your students who are interested in science and research, and try and put them in touch with scientists that have experience in the field that they're interested in. I think, in my opinion, the main way to be successful in science is to have your support system within it. So you want a mentor that can help you along the way. And Molly just asked what got me interested in endocrine disrupting chemicals. And really it was my first research experience. And so I'm just going to put my references and acknowledgements up. Thank you to the Hours office at Radford University for the funding for my very first research project. Thank you to the ecotoxicology lab and all my lab mates that helped me. Miracle Davis was one that helped clean the lab a lot, and I'm sorry I didn't help more, Miracle. Thank you to the Radford University Biology Department, because all of the professors there are super awesome and really want to help their undergraduates understand research. Thank you to the NIH and ECU for helping me expand on my research at ECU. Thank you to all my advisors, supporters, lab mates, Dr. Sarah Bryan, Dr. Jason Davis, Dr. Krista McCoy, Dr. Sarah Amato, my friends, my lab mates, and my family. And now I'll answer the question of how I got involved in endocrine disrupting chemicals. So really it was just that first project. I originally was a nursing major in college, and Dr. O'Brien, the woman that got me super into research, was my anatomy and physiology professor. And I was like, how do I, stand out in the nursing program? It was a competitive nursing program at Radford. So one of the things was, I was like, I'll get involved in research. And so next thing you know, I'm pretty deep in understanding the endocrine system. And I was like, this stuff's cool. So I changed my major to biology 2 years into my college experience and just dove into research all the way. And I did not know that research was a job that people could have. So graduating from high school and going into college, I had no idea that a biologist was a job. And so that's just another, career development learning curve that we got to start talking about more, because if you don't have those resources in high school, you're not even going to know what your options are in college. And so I was really thankful that I had the experience to receive the undergraduate fellowship, the SURF award at Radford, because that gave me the funding to complete and carry out my first project. And it gave me a lot of experience to understand how research is conducted. And it's also just really cool. Like, you spend time in the lab and you feel like you're a mad scientist as you're like in the dissecting scope, pulling out tiny little ovaries from your fish. So it just was really fun for me. What was the most interesting part of the research process for you? Understanding that everyone's confused all the time is a big one. Because I used to think everybody, like, I just had to catch up. I was playing catch up all the time. But really, this is, with all fields, it's an active field of research. It's constantly changing. We're constantly learning new things. And really, I think what I learned most at ECU was that there is so much in the genome that we don't understand that it's so hard to speak to any of it. So not only do we not know about, all of how our genes are acting all the time, we don't know how endocrine disruptors are playing a role in activating or deactivating some of these genes and what the downstream effects are of that. And there's also something called epigenetics, which are kind of on top of the genome. They're little proteins that will either turn on or off genes that exist kind of exclusively of the genome. So that's just another layer of complication that we can add to the puzzle. But it's pretty cool. I think the most interesting part is just that we're constantly expanding on it, and every bit of knowledge is good knowledge. And Molly said, but we are always concerned about chemicals for human health. But like you said, these chemicals also affect animals like our fish. So can you tell me more about how the ecosystems might be affected by EDCs? Yes. So I would say that the thing that I was worried about the most was is the population is the sex ratio going to be skewed? So are we going to see you know, less females overall, and what is that going to mean for the population? So can that specific population of fish survive the exposure? Can they survive chronic exposure? So you have to assume, you know, if you have a fish in swimming in this toxic water and it develops all these traits that you shouldn't have, such as sperm in a female, then we might see the population just start to decline just because there's less females, there's less fecund females at least. So there may be females, but they may be looking more like hermaphroditic animals. So they may produce less babies. And then we know that mosquito fish, by the name, they eat mosquito larvae. So we can assume that without mosquito fish, but you know, maybe we'll have more mosquitoes, more disease. Climate change, we see mosquitoes traveling upward and north. So it's just, there's a lot of downstream effects, and it's kind of like one thing after another. So it's like that ping pong effect. But also, what are some of the biggest challenges in explaining these scientific concepts to folks with a limited scientific background? I guess the biggest challenge is not scaring people off, because something like the proteomics pathway looks really scary. And there's all these words on here like bromobenzene 2,3-dihydrodiol. I don't know what that is, but we can, you know, look at these pathways as a whole and kind of look at the one downstream effect that it's causing, and then understand. So it's really understanding tiny, tiny little parts that come together. And another thing is the mass spectrometer is a super insane instrument. But really, it's just the learning curve of learning to operate it. Talking about these things gets easier every year, but I do feel like I'm a little rusty. I haven't done a scientific presentation in about a year until today, so that was a little tricky. And then Molly, there was one more thing I wanted to mention about the ecological effects is bioaccumulation. So we hear that buzzword a lot with microplastics. So you can see, you know, the bird will eat the fish will be exposed to microplastics, the bird will eat the fish, and then, you know, the bird has even more because they ate the fish that had it. You can see it in like whales if they eat the different animals that have the microplastics in them. And so bioaccumulation is literally what it sounds like. It's the accumulation of these toxins throughout the environment. So the bigger animals are going to have even more of them. And so that's a huge problem too. And yeah, so I think that's wrapping up a little bit of my work. Did anybody have any last minute questions? If not, I will sign off, but I also work here, so you all know where to find me if you ever want to talk about it more. There's a lot of things I didn't mention because I wanted to keep it kind of short, and I really wanted to touch on more of the techniques that we used in the lab instead of a lot of the content behind it. And also, I only worked on Krista's project for a year. Dr. Krista McCoy at ECU did a lot of her work with Dr. Sierra Amato, who is now at the NIEHS. And yeah, so I can't speak to their results too much, but the process and being involved in their process was really, really cool to watch and quite the learning experience. So I just want to thank everybody for the opportunity to talk and for listening. I've watched a lot of these, obviously, and I was really eager to try one. So thank you so much. And we will be back. Actually, tonight, we have another molecular biology talk by Cameron Sale. And we are both just bachelor's students. So another thing that we wanted to promote was that science can be done even at this lower level, where you don't have to go and get your master's or your PhD. You can still have fun and, you know, do science communication even at the lower level. So thank you all so much, and we will see you next time.